This Genetics in Society project is on RNA interference, or RNAi for short. We'll start by really quickly going over protein and protein synthesis, specifically of eukaryotes, because RNAi is based on proteins. It is also important to go over proteins because survival requires many tasks, such as ATP production, waste breakdown, growth, pretty much all the ones on this slide and more. All of these tasks require proteins. So proteins are long chains of amino acids that fold into a 3D shape to form the protein. The way it folds depends on the identity of the amino acid and their positions in the chain, but many of the ones we need are not found in the environment, so our cells must make these proteins. In order to make proteins, we have to first make RNA from the template strand of the DNA. It will be complementary to the template strand, but less stable because it's only meant to be temporary. When we are ready to make a protein, it will bind to the ribosome and begin protein synthesis. There are several types of RNA, but for this presentation we focus on messenger RNA and RNA interference. In order to make RNA, we have a process called transcription, which is when an RNA's polymerase comes in and actually binds to the DNA strand in order to synthesize RNA that is complementary to the template strand. After it's done being synthesized, it is then edited to be more stable and to remove non-coding regions, which are called introns. A cap and a tail are added so that the RNA does not get broken down before it can make its way to a ribosome and be used for protein synthesis. But not everything is going to function perfectly. There can be problems in any steps, DNA, RNA, transcription, translation. But for today, we're going to discuss viruses and the genome itself. So our first problem is viruses. And the thing about viruses is they are not considered living organisms because they cannot carry out metabolic processes and they cannot replicate on their own. So in order to do these things, a virus has to take over a host. There are a variety of ways a virus can get inside a host cell, but we have two examples here on this slide. For example, we have an enveloped virus here, which has a membrane around its viral particles, and it fuses with the cell membrane in order to end up with the virus particles on the inside of the cell. In the other example, it just goes up and it gets engulfed into the cell in a process called endocytosis. Regardless of how it gets into the cell, the virus is where it needs to be in order to use the host's machinery to replicate itself. So now that a virus has entered a cell and replicated itself, it is ready to leave. It needs to go and continue its life cycle and infect other cells. So there's a few ways it can do this, but on this slide we have two examples, some of which are more destructive than others. For example, we have the adenovirus, which once it's gotten into the cell and replicates, it leaves by popping open the cell in a process called lysis, which essentially kills the cell. This is an issue because, your cell is dead now, and if you're a multicellular organism, you have different cells devoted to different tasks, and if all the cells doing one task die, chances are you're going to die too. On the other hand, we have a process done by influenza A viruses, among other viruses, where it just buds off the membrane until the membrane pinches off, and now you have virus particles with membrane wrapped around them like this. And either way, having a virus isn't good. It doesn't matter how destructive it leaving is. Having a virus takes over your entire metabolic processes and uses your resources for itself. Either way, it's a problem and one we need to solve. Alright, and then the second problem is the genome itself. No genome is perfect and mutations may occur, either by random chance or through genetic inheritance. Some mutations may be silent, have minimal effect, or even be beneficial under certain conditions. Other times, mutations can be harmful, ranging from inconvenience to lethal. The way a mutation may affect a person depends on what the mutation is. For this presentation, we focus on mutations that lead to the overproduction and buildup of certain proteins, lead to proteins that fold incorrectly and therefore cannot function correctly, and lead to proteins that are actively detrimental to the cell. Essentially, any mutation that causes an incorrect or overabundance of protein. Before we get into an example, we will do a quick overview of simple Mendelian inheritance. So every individual is supposed to have 2 copies of each chromosome and 2 copies of each gene. So right here, these are the chromosomes and these are the genes. Copies can either be the same or different, and different versions of the same gene are alleles. So these are alleles. They are different versions of the A gene, for example. People can have extra or less copies of their chromosomes, but it typically leads to disabilities and death. Each parent passes on one of their alleles to their offspring at random. So you can see here the parent on the left passed down a big A and the parent on the right passed down a little a. Dominant alleles only require 1 copy to be expressed, and only one parent has to pass on a dominant allele for expression. So for example, both these children have one big A allele, and so they are red like their parent on the left. Recessive alleles require 2 copies to be expressed. Both parents must pass on a recessive allele for expression. So here, the parent on the left has a little allele and has passed it on to the child on the right, and so the child on the right is blue, like the parent on the right. Now that we've gone over simple Mendelian inheritance, we can talk about Huntington's disease and why it's relevant. Huntington's disease is a dominant mutation in the gene that encodes for the Huntington protein, and when you have that mutation, the extra repeats of CAG, which we'll talk about in a minute, cause your protein to fold incorrectly. And when that protein folds incorrectly, it builds up in the brain and causes brain damage and eventually death. So a normal allele for the HTT protein has around 20 repeats of the CAG trinucleotide sequence, while a mutated allele has extra repeats of the CAG, and those extra repeats are what cause the protein to misfold. The more repeats you have, the more brutal Huntington's typically is for you. So if you see in the chart, it just shows the frequency of people who have certain amount of repeat sizes. So the most common is between 16 and 19. Those would be people who do not have the mutation. And then you got people between 40 and 52 repeats, which those would be people who have Huntington's disease. In addition, number of repeats is linked to age of onset, with the more repeats equaling a younger onset age. However, while it is strongly linked, the number of repeats is an unreliable factor when determining onset age of an individual with a mutated Huntington gene who has not yet had the symptom onset. So if you see here, this is just repeat size comparing age, with more repeats being younger with the onset of Huntington's. And because Huntington's is a dominant mutation and it takes so long for someone to begin showing symptoms, if they do not know they have Huntington's, they may start an entire family, and then by the time they find out they have Huntington's, they may have kids or grandkids who are affected. And so by the time someone finds out they have Huntington's, it has gone through and it has taken an entire family tree of people who are now going to face this very serious genetic disorder. Huntington's is not alone. There are plenty of other genetic disorders that are like this or that sneak up on people through recessive inheritance. And so it is very much a problem we need to uncover. And the solution to both of these problems may be an entirely natural phenomena known as RNA interference or simply RNAi. So for problem A, we have antiviral RNAi, which is not one size fits all. Cells must develop RNAi unique to each virus, but antiviral RNAi finds viral mRNA and destroys or silences it before it can create viral proteins. So with this, viruses still get in the cell, but they do not get to make viral proteins that can continue to take over more of the cell or even destroy it through lysis. For problem B, we just have regulatory RNAi. So the genome already encodes for RNAi, and that allows cells to regulate a gene after mRNA has already been made from it. So a gene like Huntington's would create the mRNA that codes for the mutated Huntington protein, but with RNAi, the mRNA would be destroyed before it becomes a protein. Of course, Huntington's does not have a naturally occurring RNAi. Most of these genetic disorders don't, so we would have to make them. But it is a natural thing that occurs for some genes, that we create RNAi to destroy mRNA. So the way RNAi does this is through a few different types of RNA, which we will go over small interfering RNA and microRNA. So small interfering RNA comes from double-stranded RNA, and microRNA comes from pre-microRNA, which comes from primary microRNA. So as you see here, both microRNA and short interfering RNA bind with RISC, which we will talk about a little bit later, and both are post-transcriptional regulation, which means they regulate after the mRNA has been made, but before the protein is made. However, there's a few differences, being that miRNA is single-stranded, does not perfectly match with the target mRNA, and it has more variability in targets because it doesn't perfectly match. While short interfering RNA is double-stranded, it's a perfect or near perfect match with the mRNA it targets, and because it is a perfect match, it has less variability in what it can target and what it can destroy. In order to make both miRNA and siRNA, you need an enzyme called dicer, which is right here. In the case of siRNA, you need to take dicer and it will go up to the double-stranded RNA and it will cleave it into shorter siRNA pieces, which can then go and do their job. miRNA is a little bit more complicated because it has an extra step that siRNA does not. So we start with primary miRNA, which binds to an enzyme called drosha to get rid of the stem loop that is on primary mRNA, which is right here. And that results in pre-miRNA right here. So once drosha has done its job and created pre-miRNA, dicer comes in and bonds with that pre-miRNA to create an miRNA duplex. So you can see this is a duplex, not the finished miRNA. Then the miRNA duplex finds the enzyme called RISC and binds with it, which degrades the miRNA duplex into a single-stranded miRNA. Now the miRNA RISC complex is fully functional and ready to perform RNAi interference in the cell. Now that the miRNA and RISC complex is ready to complete RNA interference, the miRNA acts as sort of a map to allow the complex to find a sequence of messenger RNA that is complementary to the miRNA. It's not going to be perfect complementation, and as a result, it's only going to lead to silencing of the RNA. However, when a short interfering RNA binds with RISC, it looks for perfect complementation or near perfect complementation, which would lead to the degradation and breakdown of the RNA. So that's the difference between microRNA and short interfering RNA, is that one leads to silencing and one leads to complete degradation. It just depends on which the RISC binds with, but both will lead it to a section of mRNA that needs to be silenced or degraded based on how complementary it is. So there are several uses for RNA interference, such as research. It allows researchers to have greater control over the genes they're studying, which leads to better research and more informative research. There's also gene therapy. Its potential for treating diseases caused by proteins can give people hope where they previously had none. For example, with Huntington's disease, people now have hope for a cure from RNAi. It is currently being used in the drug patisiran to treat polyneuropathy. It was the first drug approved by the FDA that uses short interfering RNA. We can also use it for disease treatment, as antiviral RNAi may have applications in treating viral infections.